you. Yes, I didn't realize this was quite as basic science and audience as it is at the conference. So my plan for this conference, for this session, was to basically let this be audience-driven, with me having just a few slides showing. You want to get my slides up? No, wrong one. So I'm going to change a little bit, but the idea is this is your session. I've done a few trials through the years and hopefully can answer with some practical experience and some useful information to help you as you're transitioning into clinical trials. With that in mind, I'm going to skip to my next to last slide yesterday to start with today, which is on phase 1 trials and some of the problems with going from where you are into man. And then, but this was and hopefully still will be an interactive session where...
Can you point to your presentation,
yes,
sir?
Please.
Please point to it. Oh, okay, thank you. That's it? Okay, yeah, let me open that up for you.
Okay. And hopefully that will make this session worthwhile in spite of missing my target. So my first slide I wanted to talk about and use to do with today's open to stimulate some conversation is a slide about phase 1 vaccine trials. They are quite different than our typical small molecule phase 1 trial that we all know and maybe love, where people are exposed to new compounds and multiple PK samples are taken over a short period of time in an intensive environment, and appropriate safety labs are obtained, and in a couple of weeks the whole study is over and they're discharged and the data is analyzed. Well, obviously vaccine trials are quite different, and the primary focus of the first phase of this are really two. What happens with the antibody levels that we specifically are targeting are 3. What happens with any biomarkers that we're using to help us understand activity? And then 3, and probably the biggest one, is what happens with the safety. What kind of side effects are we seeing, if any? And most of the phase 1 vaccine trials that we've been involved with have been very focused on elicited side effects, having you actually go out and look for joint symptoms or ask about particular types of side effects that you may expect to see with that vaccine or have been seen with similar vaccines, and which is sort of the opposite of what we do in our typical phase 1 trial, where questions are very generalized. How are you feeling today? Anything new or different? But for the phase 1 vaccines, you're really trying to hone in on areas of particular concern, and so that frequently will be eliciting joint complaints. We'll be doing special joint exams way beyond the level of attention that we would normally give to a joint exam in our phase 1 studies. So there are some differences. We still very frequently do cohorts, and usually those represent increasing concentrations of the vaccine, but also frequently represent different additives, so that different adjuncts may be tried, or there may be, in one study we did, there were 8 different preparations that were all looked at around 4 different concentrations. So you can imagine the matrix of subject exposures and trying to figure that out with that many variables. I'm not sure that was a very good design, but that was, the company was trying to get a very early signal to move on to a 1B study with as much information as possible with as few subjects as possible. Sentinel subjects. Most vaccine studies don't really require sentinel subjects, and what I mean by that is that you'll dose a healthy, in these healthy volunteer studies, you'll dose one subject with the investigational product and one subject with placebo, so that the blind is still maintained to make sure that there's not any untoward effects that were totally unanticipated. And more than half of the vaccine studies that we've been involved with in phase 1 have not had sentinel groups. It's the technology has felt to be well enough understood that the risk of something, some sort of cytokine storm or unexpected toxic reaction like we saw with Tejanero that sort of changed our whole approach to safety a few years ago, that we don't have those concerns with most of the vaccines that we do with small molecules. And then the selection of the endpoints. As I mentioned earlier, the biggest one in most of the studies we've done has been safety, but right up there with it has been what kind of antibody response do we see, and what specific endpoints or biomarkers are we looking at. And we have had studies that have had as many as 18 unique biomarkers tied to one of these vaccine studies, trying to better understand what it was really doing. That's unusual, but you can get a lot of information in these early trials that wouldn't be available to you in a much larger study, because you have a captive audience. And but typical antibody levels are not the only endpoint that you typically look at in a phase 1 vaccine study. And then inpatient versus outpatient. Vaccine studies are not over in a couple of weeks, and most of the phase 1 study, I don't think I've done any in vaccines that have extended for less than 6 months. And I'd say the majority run a year, with the frequency of the visits and the safety checks and the antibodies or biomarker checks goes way down after the first 30, 60 days, but they're still seen monthly or bi-monthly throughout the rest of the study. And a particular attention, once again, paid to safety labs and adverse events as well as our efficacy indicators. And do you need any inpatient time at all in a typical vaccine study? And what we ask the sponsors that we work with is if we're doing lots of complex kind of determinations, particularly in an elderly population, we clearly want to keep those people in-house for at least overnight and make sure everything is going well. If it is a particularly novel type of manufacturing process or a particularly novel type of compound, then we generally prefer an inpatient stay. And we've seen vaccine studies that are totally outpatient to studies that have brought people in the night before so that you can get all the baseline labs you want and baseline assessments of vitals and everything else, and then dose them early the next morning. And then we've had them stay as long as 4 days, looking at particular markers that they were determining if there was any change or not. So there's a lot of variability there. My personal bias is if you're unsure about if it's a novel process, you're unsure about the safety, you're unsure about the population, then by all means, it's prudent to do it in an inpatient setting. Experience. As I kept talking about for the vaccine studies in general, experience is really important. These are not the simple studies that we're led to believe when different companies are budgeting with us. They actually require a level of understanding and competence that and great rapport with your subjects because they're going to be in the study for a year. So it's important that the sites that you work with understand vaccine studies, but it's even more important in the phase 1 arena where you have the multiple biomarkers, generally some inpatient stay, a lot of assessments, and you want to watch these subjects closely for any changes, any signals, something that might help you better with the further development of your vaccine. So if it is important for routine vaccine sites to be experienced, it's important for phase 1 vaccine sites to be triple experienced. It's really critical to good study. And then match your site and your population that you're working with. You really don't want to, if you're doing a vaccine in elderlies, then you probably don't want to work with a phase 1 unit that basically does healthy volunteer studies in the 45 and younger. You want a phase 1 unit that does patient-based studies, that has lots of experience with elderly subjects, and the vast majority of the vaccine studies that we are involved in all want at least a certain percentage of their subjects to be in an older age group, because for so many of the vaccines, they're the targets. And then communication. And in phase 1 vaccines, it's critical that the medical monitor, the project manager, the clinical scientists that actually understand the compound, the PK people that are looking at this, that everybody communicate frequent and often from the early planning stages all the way through the final patient visits and database lock. So it is of all the things I've said, communication in this arena, just like experience, is critical. And whether you're an emailer or you're verbal like I am, frequent communication is essential. So I've given you my biases about this transition. Some other points that are here is I don't think, I can't think off the top of my head a phase 1 vaccine study that we have been involved with where everything happened on time. FDA delays, questions are exceedingly common, and we're all very optimistic. Oh no, they're going to love this. They like our design. They're going to love our compound. And then first response is a 17-page document that it takes a month to get all the answers to. And then after the responses go in, it's another month, and another 5-page document now comes back with questions. So it plan for delays. They're even more common than they are in the later phase studies, and it are manufacturing issues. Most timelines for phase 1 vaccine trials are based on the fact that their manufacturing process is going to be okay, that it's going to pass all of its standards. But this testing is ongoing while you're setting up your trial, and your trial is sort of set, okay, the day we get the results from this, then a week later we're going to be able to go ahead. But the results aren't always what you expected. So it's important to think about all the things that can change, not just from the scientific point of view, but from the practical point of view, getting you from the lab into people. So I've sort of monopolized what was supposed to be an interactive talk and my plan is to go through some of these slides and give you different ideas and perspectives than are additional than what I did yesterday. But what I really need is some of your participation and questions. So, yes, sir. Thank you.
Speaking to the microphone, I don't have any clue how this clinical trial is conducted. So the question I have is really from the very beginning. My first question is, are you interested in doing clinical trial? If people in the audience have a product, and how do we get started? If you can start from the very beginning, it will be very helpful to me. I don't know, maybe the other individuals in the audience are very knowledgeable, so this may not be something practical to do. That's up to the chair. Thank you.
Well, obviously, this is what I do for a living, is phase 1 work and taking compounds into man. So I'll put a few of my cards on the table back there at the back for anyone that would like to talk further. But the process generally is longer. Don't get me started on how I think the process should be, because it could be a whole lot quicker, simpler, and cheaper. But nobody asked me, so. The process involves what you and the audience are doing, which is developing new compounds and new approaches that look promising, and then you're doing your animal testing that gives further ideas, and you have manufacturing processes that you're probably doing with a partner that has done this previously and is actually developing the compound. So that once you've got a compound that you can make reproducibly, then you've got to do animal testing that's going to tell you about safety, and depending on the urgency of the vaccine. For the Ebola vaccine that we were fortunate to get to work with, the FDA and the European authorities allowed all kinds of shortcuts to get this into man. A lot of you've got to show something in the animal that suggests that you've got an appropriate antibody or biomarker or some kind of response. You've got to, and that they made them do, but and a certain amount of safety. But a lot of the congenital anomaly studies, the teratogenicity, a lot of the other work, a lot of the stability data that they're going to make you do on your vaccine, they just skipped all that, and it was done, but it was ongoing, and they moved on into phase 1. But basically, you've got to show that your compound's stable. You've got to show that it, in at least 2 species, that it doesn't have a lot of toxicity, and that's going to be done both grossly but also anatomically. And there are well-defined protocols for what you have to do to get to going into man. And then once you've got this packet of information, you communicate with the regulatory authorities, and you submit what's called an IND and investigate new drug, and you you present them with the protocol and a plan for the development of your vaccine, and they respond with alterations in your plan, comments about the science, as well as the operational issues around your protocol.
But the IND is needed in the U.S.
The IND is U.S. It's not, no, but in the EU, they have a comparable approval. It's not called an IND, but it's the same thing. So, and I didn't say this, but sometimes it's faster to get into man in Europe than it is in the U.S. Sometimes it's faster to get into man in the U.S. than it is in Europe. And a lot of big pharma will submit regulatory paperwork to both systems, and they'll pick the one they like the best, and that's where they'll do their first in man.
We're running out of time, but I think it's a very useful discussion. And I'd like to ask you a question, and if possible, a short answer. So when I look at it, so if you, based on your extensive experience running clinical trials, what are if you could tell us 2 common mistakes made by the sponsor and 2 common mistakes that should be avoided by the center that actually running clinical trials.
Okay.
And what would those be?
Okay, far and away the most common mistake has to do around trying to do things before they're ready to do it on the sponsor side. Like I said on my first slide yesterday, does it really matter if you do a vaccine study in 4 weeks versus 1 week? Particularly since it's going to run a year, and when you get to phase 3, it's going to have huge numbers of patients. And if that lets you get the best possible study, then why would you do anything else? In a lot of our projects, we like to do a day or 2 where we do a small number of subjects, and then we sit down at the end of the day and debrief the staff about any problems they had, any issues, and then the second day it goes a whole lot smoother. And we sit down at the end of the second day when we're still doing a small number and debrief the staff and fine-tune our process for that particular study, and then go to the large numbers. So being in too big a hurry. And I also mean that in terms of what they're expecting from regulatory, and something where it always comes up are flu studies. Everybody starts planning a flu study to happen in September. Well, that's great, but then they don't have everything is planned to be ready in September. So if you're going to do a vaccine study that has a seasonal component, and you know that people are going to be either vaccinated with other vaccines, or that you've got a limited window of which the disease has a high incidence, then give yourself a month or 2 before that to have all your vendors ready, to have your sites ready, to have everything ready so that you can get started on time. I can't tell you the difficulty it presents for the sponsors and the sites for flu vaccines in particular to try to start a flu vaccine study in November.
All right, I think very quick question.
Sure.
At preclinical trials setting, when you use 2 animals, is it more or less mandatory to use macaques? Especially on HIV, everybody uses macaque. Is that mandatory? I mean, with T-cell immunity, macaques have horrible MHC, and you don't want to use them.
I am not expert enough in the preclinical arena to tell you that for sure. They're in an awful lot of the investigator brochures that we read as we're looking at their development, but that's
how many people have used humanized mice as a preclinical model?
I don't know.
Okay, sorry.
I'm sorry. Most of the preclinical is my exposure is it comes to me as a packet of information to review as and make sure that I'm comfortable with it before giving it to my phase 1 subjects.
Well, let me ask the audience, has anybody used humanized mice in their studies, and is that you think, because this discussion comes up pretty often, do we really need to go into non-human primates? Is a pre-humanized mouse model is the right model for us, to move forward with whatever vaccine product that we have? Anybody has experience with humanized mouse model? All right, I guess nobody. But it's actually a serious consideration, right? Especially given the problems in Europe, it's almost impossible, to work with non-human primates. And there are pretty good humanized mouse model actually being developed. And indeed, we just established a humanized mouse model in our program, and hopefully there is a good one for HIV, by the way. So I mean, there are too many of them out there, but still, I mean, it's difficult really to know exactly what model will be appropriate for your particular study. And it's amazing, just from my own perspective, we still have a couple of minutes. When before we established the model, I had to talk to Jackson Laboratories as well as Taconic. And it was amazing when you talk to the scientists who are developing these models, and you have to talk to both of them, by the way. And as you dig deep into the system and you realize there's so many variations, there's so many things that you have to take into consideration as to how those models actually behave. With respect to their B-cell responses, for example, some of those models, they don't generate a B-cell response. With respect to, of course, T-cell responses, you have a xenogenic system in a sense, right? It depends whether you're doing this BLT, the liver thymus and all that. And in that case, you still have, the issue of, okay, what is the repertoire of your T cells? And have you really changed it, right? Because now you're having some of the T cells will go through the mouse thymus. And so there are a lot of whole array of issues that to consider. But I think at the end of the day, hopefully we will come up with something that will be as predictive as non-human primates. And they're not always predictive, by the way, right? There are a lot of examples of that. Okay, with that, I appreciate Dr. Smith. It was a great presentation, and we'll have our next speaker. I think I have one more. He's taking a picture yesterday. I guess we don't need, do we need to take another picture?
We don't need to do it yesterday.
Okay, thank you very much.
And I'll stick some of these cards on the table back there in the corner, and anybody that's got questions or things about going into trials, feel free to call.